Hello, everybody. My name is Bryan Bell, and I'm the Senior Market Strategy Manager for Biomarker Discoveries at Takara Bio USA. I appreciate you joining us today. The title of the talk is Uncovering Biological Insights Through Enhanced Biomarker Discovery. Over the last 10 years, a number of sequencing advancements have enabled researchers to do a better job in discovering and validating biomarkers. Now, improved biomarker discovery is needed to fully realize the potential of precision medicine. And advancements in genomics and transcriptomics are already leading to great discoveries of clinically relevant biomarkers, but there is still work to be done. There are a number of different methods for doing transcriptomic analysis, whether you're looking at bulk sequencing methods or single sequencing methods, but single sequencing methods have a number of advantages over bulk methods. For instance, single cell analysis allows for a deeper understanding of individual cells and their molecular characterization in complex human disease states. It also allows you to do analysis of minority subpopulations or those rare cell types that are of interest. And lastly, single cell analysis allows for discovery of rare biomarkers that are often masked by bulk sequencing methods. However, there are some current limitations and challenges that are present in the single cell workflows. Some of these are manual processes are still part of the workflow. Miniaturization can be difficult and not easy to achieve. And lastly, a number of the workflows for single cell analysis currently limit transcript information to just the ends of the transcript. This highlights the need for capturing full-length information when discovering biomarkers. Some of the benefits of looking at full-length mRNA-seq data are it allows you to do a whole transcriptome interrogation of each individual cell. It gives you the ability to identify splice variants, gene fusions, and SNPs, which can be clinically relevant. And lastly, it can give you a better understanding of your precious samples, where this added information can be of benefit. And today I'm very excited to introduce the new SMART-Seq Pro for the Isolate CX, which is going to allow you to uncover new biomarkers through discovery of novel isoforms, fusions, and SNPs using the complete solution for automated full-length single-cell mRNA-seq. When we talk about a complete solution at Takara Bio USA, we talk about from sample all the way through data analysis. And our very powerful Cogent analysis pipeline and our Cogent DS software is included with our Isolate CX system, allowing you to go from sample to sequencing to analysis, all from the same vendor. And when we talk about capturing more information on the single cell level, this data here just highlights the read depth versus sensitivity when looking at sample types such as PBMCs. You can see from the commonly used 10x genomics platform that's looking at the 3' end of the transcript, after about 50,000 reads, you've maximized the number of reads or genes you can identify with this technology. However, with SMART-Seq Pro for the Isolate CX, you can see this is a much more sensitive technology, and as you sequence to a greater and greater depth, for instance from 50K to 100K to all the reads in the sequencing lane, you can see you can detect more and more genes. And this is mirrored similarly with the number of transcripts. Again, the 10x genomics platform really tops out about 50,000 reads, but with SMART-Seq Pro for the Isolate CX, a much more sensitive full-length information platform, you can see you can get more and more information in order to meet your needs. But it's not just sensitivity in the number of genes you detect. There's a lot of information in the full transcript that you're just not going to get with end counting methods. For instance, the clinically relevant BCR-ABL fusion gene, which you can see pictured here, where the BCR gene is then fused to the ABL gene, can only be detected when looking at a full-length sequencing platform such as the SMART-Seq Pro for the Isolate CX, where 85% of the over 1,100 cells analyzed showed the BCR-ABL transcript. While looking at the end counting method with 10x genomics, this fusion protein, which is very clinically relevant, was not detected. And it's not just isoforms and fusions that are detected with a full-length transcriptome analysis, it's also the number of SNPs that you can identify, which is really important when you're trying to discover novel biomarkers. And as you can see in the top graph here, where we look at the normalized transcript position from the 5' end to the 3' end, comparing Takara Bio's full-length solution with SMART-Seq Pro for the Isolate CX versus the 3' end counting method from 10x genomics. This is graphed against the clinically relevant SNPs that are identified by the ClinVar database. And you can see you get much better coverage across the full-length transcriptome with SMART-Seq Pro for the Isolate CX, while you're missing a majority of the clinically relevant SNPs with the 10x genomics 3' end counting. And in fact, when you actually look at the data, you can see about a 5 to 10-fold increase in the number of clinically relevant SNPs identified with SMART-Seq Pro versus the 10x Chromium. So what we're showing today is that you can move beyond the current limitations of single-cell RNA-seq technologies. There's no more compromising when discovering biomarkers. You can exceed the sensitivity of plate-based methods, and you can get full-length information that you miss on droplet platforms. So just to reiterate, SMART-Seq Pro for the Isolate CX offers a complete solution from sample to data analysis. It maximizes data from each individual cell, and it uncovers novel biomarkers that are missed with other technologies. And that's good science.